Good morning, everyone. Today we're going to discuss the stem cells in the nervous system. So in our book, we have the description of stem cells not only in the central nervous system, but also some stem cells in sensory organs. So have a lot of content in there. But due to the limit of time, I'm going to only cover the cells in the stem cells in the central nervous system, including the stem cells in the brain, as well as the stem cells in the spinal cord. But to start the lecture, I would like to describe the many cell types that we found in the nervous system. So first of all, the most important cell type in the nervous system would be the neuron, right? The neuron is the cell that will give out command, right? Give out orders. So they do that by sending usually neurotransmitters through the structure called synapse. You probably learned that elsewhere in maybe biology or anatomy. The neurons can be divided to three types. One is motor neuron, the other is sensory neuron, and the third is interneuron. The motor neuron will send out orders for action, right? Such as making the muscle to contract, make gland to secrete hormones. And, that's basically it, sending out orders by sending out neuron transmitters. That's what motor neuron does. And then sensory neuron are receptors, like, we smell things, we see things, sensory neurons will be involved. And then the third type is called interneuron. By that name, that's the neurons that make connections between neurons, right? You probably have motor neuron or sensory neuron at the terminal. And now in the middle of that chain, you have interneurons. Okay, you have three types of neurons in the nervous system. They perform different functions. But although neuron is the most important, we still have some non-neuron cells in the nervous system. We call those cells glial cells. By the name, it describes that those cells will be kind of gel-like, right? To glue-like, to provide a gel-like structure in the system. The most important ones will be the oligodendrocyte and astrocyte. So shown in this picture, oligodendrocyte is the ones that's shown as pale blue. The pale blue ones are oligodendrocyte. Oligodendrocyte secrete myelin. So they will form the structure called a sheath along the protruding part of the neurons. The neuron here is labeled as yellow, while the oligodendrocyte is pale blue. The structure there, you see the sheath around the axon of the neurons are produced by oligodendrocytes. So the function of oligodendrocytes is to insulate the axons or dendrites, and then make the signal transmission to be more efficient, faster. Okay, they increase the efficiency of signal transmission. So that's the function of oligodendrocyte. The third, I'm talking about the cells in the order of their importance in the nervous system. First is neuron, the second is oligodendrocyte, and the third will be astrocytes. The astrocytes literally means they look star-like, right? The purple cells shown here is the astrocyte. Their function are kind of, a lot of functions can be assigned to astrocytes, such as helping the neuron in their metabolism, providing support, structural support, and the nutritional support and such. So they have the most diverse function in the nervous system. So we will omit that part. So astrocyte also is the most numerous. You have the most astrocytes when you compare to other cell types in the nervous system. And then that's the three most important: neurons, oligodendrocytes, and astrocytes. And in addition to the main cell types, we found other cell types such as microglia. Microglia shown in this image here, illustrated as red. They are not actually originated from the ectoderm. They are originated from the hematopoietic system. So they are the immune cells in the brain. Yeah, elsewhere, if you found similar cells elsewhere, you will call them macrophages. But here, they're called microglia. These microglia cells, since they are similar to macrophage, their function will be fighting foreign substances, pathogens, and such. So they're actually essentially the immune cells in the central nervous system. And then the last one I want to describe is called ependymal cells. Ependymal cells, they are another glial cell type, but they are similar to endothelial cells that you found in blood vessels. They're endothelial cells, reminiscent of endothelial cells, not exactly the same. Okay, they will line the cavities in the brain. They will call the cavities ventricles, right? They will line the ventricles. Therefore, apparently, they're separating, the spinal cord, the spinal fluid, brain fluid there from the structure. So they create that cavity. They are lining the cavity, and apparently they can be used for factor exchange between the fluid and the rest of the cells, right? So they're separating the fluid and the structures. So they're important in relaying signals, because in the fluid there is going to be some growth factors. Those growth factors, if we're focusing on stem cells, those factors will be important to keep the stem cells in their stem cell state, or cause them to differentiate depending on the situation. So these are the 5 cells we described, the 5 most important cells in the nervous system. And now I move on to the next part. I would like to introduce to you this pioneer in neural stem cell research, whose name is Joseph Altman. Joseph Altman was first a scientist in MIT. While he was there, he was an assistant professor, started his career in MIT, and he made some spectacular discovery. When we review history in today's term, we feel like he was the founding father of neuron stem cell, or neuron, the adult stem cells in the brain. What he did was that he followed the work by Charles Philippe Leblanc. By this point, I hope you have heard from me multiple times of my description of the work by LeBlanc, and he used the radioisotope to label DNA within different organs, so he can distinguish the proliferating cells, right? Because he put H3, thymidine into the organ, and then those will take the 3H thymidine, and then once the cell will proliferate, he can track those cells. He can track the proliferating cells because those cells will have the 3H thymidine. So LeBlanc described that he was the one who identified who proposed the stem cell zone theory in the small intestine stem cells, right? He was right, and later on he was proven to be right, and he also described some part of a hematopoietic stem cell as well as satellite cell. So LeBlanc made a lot of contributions early on, and this scientist Altman just followed the work in brain, and he found there are proliferating cells in the brain. And later on, he was even able to describe that in different regions of the brain, the activity of proliferation is different, and he was able to track those proliferating cells along some path, because during cell differentiation, if those cells are stem cells, they will not restrict themselves to one place. They will migrate to different areas of the brain, as you will see later on. That will be the case. When cells differentiate, they actually migrate. Okay, so he made a spectacular discovery, but it was very unfortunate that at that time, nobody believes that in the adult brain there is stem cells, because just as in the heart, when people will have an injury in the brain, they will almost never recover. No matter if it's a stroke or it is a traumatic injury, usually that means loss of function permanently. People get paralyzed, right? So nobody believes that, especially the big wigs at that time. They just feel like he said something that's nonsense. So he wasn't able to get his project funded by the government, and MIT even didn't give his tenure. I don't know if you guys know the tenure system. As an assistant professor, 5-6 years later, you will get evaluated. If you will pass the evaluation, you get tenure. That means you're going to be a tenured professor. That means you're going to be a lifetime professor in a certain institution. But if your performance is not satisfactory, they will dismiss you. So you have to go. You will lose the job. So that was what happened to this great scientist, Joseph Altman. He was able to get his tenure. He had to move on to a smaller institution, not really small. He went on to Purdue University, but he couldn't, continue that line of research. Of course, he did some more, but the majority of his later work is conventional developmental, biology, not much related to his earlier work. And he still couldn't get himself funded, and he had to draw brain atlas to get money to support his research. It was really, really unfortunate. And later on, I believe it was in about 1980 or so, another scientist described neuron stem cells in bird. And the evidence was very, very strong. A lot of people believed that there is stem cells in the brain, but another scientist, I don't remember the name. It's another, I don't want to use great. It's like a well-established scientist in the field, published a very influential paper and said the neurogenesis is only restricted to lower species, not relevant to human. In humans, you don't have it. And then that paper kind of blocked this field for another 20 years. So only in early 2020, and people start to build more evidence, and now it's widely accepted that there are neuron stem cells there in the brain, in human brain. So as you will see today, I'm going to explain to you those the current knowledge, but it was really unfortunate that in 1960s somebody already described that, but it's being blocked by the people in the field. They just don't recognize that. Although the evidence was, at early days, no work is perfect, limited by the technology, right? And so it just blocked that possibility and didn't allow the research funds to flow into that important area of research. So you see that being a scientist sometimes are not very easy. You could be the Piero Anversa, like I described in the previous lecture. He was at the top of the field. People say he was the godfather of heart stem cells, but it turned out he fabricated a lot of his results. So he kind of damaged the field in a very severe fashion. But this scientist, he was right, and he, described honest work and couldn't get that funded. It's really unfortunate. I guess what I'm trying to say is, doing research, usually you will be driven by two reasons. One's out of curiosity. You just want to tell what is the truth, how things work, especially in live animals, in humans. You want to tell people how things are organized, how signals are transducted, and how things work. So that could be your drive, or some other times for other scientists, they're driven by whether they can make something useful, right? Like make a drug, produce a drug that can be used for treating some diseases. Primarily, I see people have these two different kinds of drive. But I would say maybe the first one is more important. So your curiosity, your desire to pursue truth is more important, because in that case, your satisfaction is only from, the ability that you can produce the truth, or the outcome that you consider is closest to truth, right? You are not satisfied with anything else. But if you're driven by whether it is useful, sometimes it could be misleading, right? Because you will be competing with a lot of others. You try to say, my method is useful, my theory is useful, yours is not. That sometimes could lead to a lot of bias. So, but as long as you have either of those drives, that is still a good thing, right? Like Piero Anversa, I don't know, maybe he's driven by pursuing fame, and then he doesn't care about truth and then start to fabricate things. Okay, that's a little bit history. And now I will move on to the description of the two regions that Altman helped to find. Of course, at that time, it was not that clear, but as I said, as I described, that early on, he just used the radioisotope labeling to track the proliferating cells, to track their path. And at the beginning, he found that there was a region called the dentate gyrus. The dentate gyrus is a place, as shown in this picture. It is this DG, dentate gyrus, is a structure within the hippocampus. The hippocampus is a region that is responsible for emotion, memory, and automatic nervous system, right? It's mainly about emotion and memory, and automatic nervous system. So within that hippocampus, there is this special structure called dentate gyrus. That's where he first identified the proliferative activity, and he felt that there may be progenitors or stem cells located in that region. But he wasn't able to prove the stem cells in that part, but instead later on, he found that in another place that is called the ventricular subventricular zone, shown here. If you can see my pointer showing in this place, it's called the subventricular zone. That is because you have ventricle here, and this is regions close to that ventricle. It is called subventricular zone, and he was able to find that the cells here can differentiate and then move to the olfactory bulb. That's another region of the brain. So the cells are originally located in the subventricular zone, but then they will differentiate and also move along this path. This path is called rostral migratory stream, RMS, and eventually move on to the olfactory bulb. That bulb is related to learning and such. So the cells, the stem cells move quite a bit of path distance toward a different region. And he was able to identify these two places. And then later on, people, that is about the late 1990s, we start to have new methods such as immunofluorescence analysis, confocal microscopy, especially the BRDU labeling, because BRDU labeling is easier. It's easier, and also you can track by doing antibody staining. So basically, in Altman's days, he used the radioisotope, but how you can see radioisotope, right? You have to use a film. You have to use a film, and then the resolution may not be that great. You compare your film toward the structure, the anatomic structure, then you kind of deduce where those cells are. It's not very accurate because it's just the resolution is not high. So it's kind of difficult. But if you use BRDU is a nucleotide analog. It's just in a very similar fashion as thymidine. So just to use BRDU and get BRDU incorporated into DNA, and now you can use antibody to detect where those BRDU labeled DNA are, and now you have a better resolution. If you put the section under microscope, you can describe where the proliferating cells are. So with those technologies, and now now it's a consensus that in the brain you have these two major regions. One's in the hippocampus. That's the in the old days, people still describe that as the dentate gyrus neural stem cells in the hippocampus. Later on, you're going to see another name, more precise name for that. And then the other place will be the ventricular subventricular zone. It's called ventricular subventricular zone. So for either case, I would say the neuron stem cells are the function of neuron stem cells are kind of different from the other adult stem cells we have described so far, because no matter it is the stem cells in the small intestine or hematopoietic stem cells, their function is to keep tissue homeostasis, right? So you have some tissue removed because they're damaged, or it's just the way things work. They have to be replaced, right? That's called tissue homeostasis or repair. Most times the adult stem cells perform that function, but in the brain, it seems the stem cells perform a different function. It's not really about homeostasis or repair. Because as I mentioned at the beginning, if there's a stroke, and usually the function will not be restored. The repairing function of the stem cells, neuron stem cells, seems to be limited. Just like if we have stem cells in the heart, their repairing function is not that great. It's not sufficient, but they do perform a function that's just as important. So they don't create. They perform their function by creating new contact with the existing circuitry. So that is super important for learning, memory, and things like that, right? So it's safe to say that if you have if your stem cell function, the adult stem cell function in your brain is great. That means maybe you're better prepared in learning, right? Because your brain will be able to build a lot of new connections, building a lot of new circuits. So at least at this moment, people feel like the main function is memory, learning, and things like this for the adult stem cells in the brain. It's not for repairing damages. So the previous slide would describe the two zones, and then from this point on, I'm going to describe the oh no, not on this slide. From next slide on, I'm going to describe the two zones. But on this slide, I just want to tell you the method used for stem cell isolation from adult brain. So it was first isolated in 1992 by Reynolds and West, and the method they used was not, as what we have learned previously, such as FACS sorting or FACS sorting by surface markers, like in hematopoietic system, or lineage tracking, like LGR5 positive cells in the small intestine. You can label the cell and isolate the cell that way. So you learn that in other systems, but here, when they start their work, what they did is based on the ability of the cells to form a structure called neurosphere. So let me describe their procedure. So basically, they take because it's known that the subventricular zone will have stem cells, right? At least, those cells will have some proliferative ability. In the brains, you will hardly find neuron-like cells have that ability, proliferative ability. So they take that region out, dissect the region, and use some enzyme to make a single cell suspension, like you did elsewhere for other tissues. And then they put the cells in culture with a special culture medium. But in this medium, the important part is that includes EGF, this epithelial or epithelial growth factor, or epidermal growth factor, and also basic FGF, it's fibroblast growth factor, or sometimes this is also called FGF2. It's either basic FGF or FGF2, the same thing. So with the supplement of these two growth factors, and these cells will be able to form balls, form cell balls, and they call this neurospheres. And later on, they found these neurospheres will be able to self-renew. So you can dissociate this ball and make a single cell suspension again, and then those cells will be able to form more balls, more neurospheres. Okay, and that means they're self-renewing themselves. And then if you put them into a different condition, like you make them to attach to a surface, now they are not in suspension culture. They are not forming spheres. Now you put them, you plate them so they can attach to the surface, and then you can change the culture condition to include additional growth factors. And now they observed that these cells, the neurospheres, can differentiate into neurons. Shown here, this light green one, you see the axon there. The dendrites, this is a neuron, and then astrocyte and oligodendrocytes. So the three main cell types in the brain or in the nervous system, you can produce all of them through a neurosphere. So that just demonstrated, okay, you do have cells that can self-renew and differentiate, at least in vitro. And then, of course, you can further refine. And now you can further refine and see if you can find some markers, to further divide the cells that can make these neuron spheres to different subpopulations and see which one have the highest potency to generate neuron spheres, right? And then you can pinpoint to an even more defined population that give rise to this neuron sphere and eventually to 3 types of cells in the nervous system. So this is the work they have done. And then this method, sphere formation, has become a method of choice for other organs as well, such as in the heart. People will make a single cell suspension, and then try to see what cells can form a sphere, and then use that as a method of identifying potentially stem cells from the heart. So that's just an example. The method could be tried in other systems as well. So we described the many ways to identify stem cells. So you can use surface markers, linear tracking, sphere formation, SP cells, the side population cells, or sometimes you can use the different metabolism of the cell, like alcohol dehydrogenase. Maybe because the stem cell prefer to use glycolysis, and then they probably are positive for alcohol dehydrogenase, which is we say the 11th step of glycolysis. It has to complete the cycle, right, to return the reducing power and the oxidizing power to the original form. So that is actually essential for glycolysis. So some stem cell will use enzymes in glycolysis. So the positivity for those enzymes can be used for stem cell isolation as well. So it just happened in the nervous system. The first time when people isolate, they use the neuron sphere formation. And then on this slide, I would like to describe the hierarchy of neuron stem cell differentiation and their markers. So basically, you have neural stem cells in 2 regions of the brain, and then usually they will undergo symmetrical division to give rise to 2 neuron stem cells, right? That's called self-renewal. But if the situation now calls for differentiation, now the situation calls for more neuron cells. So the cell will undergo asymmetrical division. They will give rise to 1 stem cell, that's self-renewal part of this asymmetrical division. And at the same time, they generate a progenitor cell, neuron progenitor cell. Once they have this neuron progenitor cell, the neuron progenitor cell will, at this stage, you call it transient amplifying cell. It serves as a step to amplify the numbers of differentiated cells that you can generate from 1 stem cell. So this is called transient amplifying stage. But at that transient amplifying stage, a lot of times the cells will be committed. Like in neuron stem cells, once they're into that transient amplifying stage, some cells will become neuroblast, and some other cells will become glioblast. The neuroblast, of course, later on will give rise to neuron, while the glioblast will give out glial cells. The glial cells, the main glial cell types will be astrocyte and oligodendrocyte, right? So on this table, I show some of the markers people use to describe and identify the cells in this hierarchy, including stem cells, progenitors, and specific progenitors, like astrocyte progenitors, oligodendrocyte progenitors, and such. So it's a very useful table. If you're interested in this line of research, when you read articles, you don't know what those markers are, you can easily come back to this table and tell what these markers are specific for. I just want to point out some of those names so that we can have another chance to review something we have already learned. Like CD133 is called a prominent. It is a very common marker for adult stem cells, not only here, elsewhere as well. SOX2, you see, it is a Yamanaka factor. It's used for keep pluripotency, but we in one of the student-led discussion as well as in my lecture, we know that OCT4, SOX2 are pluripotent transcription factors, but they have bias. Like OCT4 will push the cells toward mesendoderm or SOC2 will push the cells into ectoderm. And here you see the stem cells specifically express SOC2. I hope that's not very surprising, right? And then I'll jump to the last one, ABCG2. ABCG2 is a channel protein. It's a channel protein responsible for couldn't remember exactly what it is used for, but it is a channel protein. And then you recall that site population is a population of cells that can export a dye, right? That can export the Hooke's dye, and this channel is responsible for that. So if the cell is positive for ABCG2, and you can predict maybe in flow cytometry, this cell will show the site population phenotype. Okay, that's just something I want to add there to review what we have learned. You know, the markers will jump out again and again. It gets shared because stem cells have some common properties in terms of metabolism and their niche. So it's not surprising that in different organs that the stem cells have some common properties. So now I move on to describe the two zones, the two stem cell regions. The first will be the so-called ventricular, subventricular neural stem cell niche. I just point to you where it is. So this is a different section. It's just showing the ventricle. The ventricle is a cavity there. And then you have this region very close to that cavity. This is called the ventricular subventricular zone. And in this zone, people have identified a cell that they call B cell. It's not the lymphocyte, okay? It's just when the author first published this paper, how they named the cells they have identified. They call this the B cell in the neural NSC niche. And then this is actually quite powerful, the B cell, because they can use lifetime, it's not per day. Lifetime, they can use as many as 10,000 differentiated neurons in young adults, not in old ones. In old ones, the number will drop dramatically. The young adults, you can get 10,000 of cells out of just one B cell. And then the B cell will have a hierarchy, you know, in terms of differentiation. The B cell will give rise to C cell. The C cell serves as the transit amplifying cell in this hierarchy. So basically, they will divide a few times, 3 to 4 times to be exact. Then they will give rise to A cell. So from B to C to A. So following this differentiation hierarchy, the A cells are now called neuroblast. They will further proliferate a few times, and then these cells will migrate along that path. Remember, at the beginning, we described a path called RMS, and they will migrate along that path and eventually reach the olfactory bulb as a different anatomical region in the brain. And then once they're there, you know, you have a chain, a cell chain. During cell migration, you have a continuous cell movement from that stem cell niche toward the olfactory bulb. You have a chain there, sort of a cell-formed chain there. And then once the cell reach the olfactory bulb, they will get detached from that chain and then move to other places, the adjacent places, and finalize their terminal differentiation and become neurons. Okay, in the illustration here, you don't see that migration. You just see the hierarchy. The B cell becomes C cell, the C cell becomes A cell. The A cell is neuroblast. Eventually, the A cell become the terminally differentiated cells. And then here I also want to describe the other, you know, the non-neuron cells within this niche. So we say, you know, the scientists divided the B cells to 3 parts, 1, 2, 3. So this 1, 2, 3 is just a different part of the the B cell. So apparently, region 2 is the region where B cell will be in contact with A and C. So the A and C cell, those are the progenitor cell, will give feedback to the B cell to tell the B cell whether to proliferate more or stop proliferating, or differentiate it or keep your stem cell status to give feedback to the B cell. And you see that the B cell have this so-called basal process. That is the part 3. It will be in contact to blood vessels. That's how the B cell will take nutrient and oxygen. So they are in contact through this region 3 toward blood supply. And then from the other end, it has something called apical process. The apical process will be in contact with a cell called E cell. E is the ependymal cell. Ependymal cell is the lining of that ventricle. Okay, so the lining of the ventricle, therefore here, not only here is where the cell will receive signals, growth factors from the ventricle, from the cerebral and spinal fluid. And the growth factors here will give guidance to the B cell, again, to maintain the stem cell status or differentiate. Okay, so this niche is very well defined, not like in the cardiovascular research, the niche is hardly defined at all. So although there are only 20 years or so of work in the neuron stem cell field, essentially, but the picture is very clear. How the cells work in these niches are very, very clear. So on this slide, I'm going to tell you the other region. This is called, pay attention to this. Now, the other region is not called the dentate gyrus any longer. This is called the subgranular zone. Why? It's actually still the same thing. It is because later on, with more work, people have identified that this subventricular zone is located in the still hippocampus, but at the interface of the hilus and the dentate gyrus. So the early work said that the stem cell is in the dentate gyrus, but later on, it decided it is, in the interface between the dentate gyrus and the hilus. So that's where it is. So that's the structure of the hippocampus, and then that structure there in the middle is the dentate gyrus, and then underneath it is the hilus. Okay, so that's where you will find it. I don't know whether it's, but you were in class, you have learned anatomy or not, but it's not the purpose for our class. As long as you remember there are 2 zones, that's sufficient. But here we have a detailed description of the stem cell niche within subgranular zone. And here, let me say something I forgot to say from in the previous slide here. So see, in other organs, when we describe a stem cell, usually the stem cell is a tiny cell. You have a big nucleus to cytoplasma ratio, right? Because the stem cell won't perform much function. So you need a big nucleus, so the genetic material is there. When it's ready to proliferate, it can proliferate. You don't need a lot of cytoplasm. But here, look at the neuron stem cell. They're relatively bigger, or shall I say much bigger than the transit amplifying cell. So they're quite big. They can be even divided to several regions, just one cell. And here, same thing again. You see this stem cell here. The stem cell identified is called radioglia-like cell. Don't get misled by the name. This is named glia-like cell. It's glia-like, but they're not actually glial cell, right? Because they are neurons, stem cells. So the radioglia-like cell is the stem cell in this subgranular zone niche. But look at the shape of the cell. It is very irregular. The previous stem cell, the B cells, already very irregular if you look at the shape. And now the stem cell here is even more strange looking. You have a lot of dendrites, right? A lot of protruding part process from the cell, a lot. So the cell is the cell shape is very irregular and also big. And now when they become the progenitor cell, that is called IPC1, that is intermediate precursors, or IPC. That's the progenitor cell. Now the progenitor cell, the immediate progenitor cell is not in the GCL, that is the granule cell layer. It is actually in the hilus. The progenitor cell is in the hilus, but once this progenitor cell will proliferate and differentiate, they move back to the subgranular zone. And then they further move into the granular cell zone, the granular cell layer. And then eventually they are not in contact with the stem cell any longer, and then they become the neural progenitor, which is called the GC cell. It's the immature granule cell. And then they are going to have dendrite and the axon. The axon will come back to the hilus because the axon now will make new connections. They will make a new circuit some of the toward some of the cells in the hilus. So that's the function of this newly formed terminally differentiated cell from the RA cell. So this niche is also well defined. And here the RA cell actually is very powerful because in young adults, roughly 9,000 new neuron cells can be generated per day within this subgranular zone. So this actually produces a lot of neuron cells. So it's amazing that in early days this get missed and people don't recognize that. And now we know that actually there are a lot of cells that get replaced every day. Okay, that's the two main zones. Remember the names and some knowledge about the differentiation of those cells. And with the last 2 to 3 slides, I'm going to describe to you the stem cells in the spinal cord. Just like the stem cells in the brain, people don't recognize that there are stem cells in the spinal cord. It's easy to understand why, because damage, injuries in the spinal cord cannot be repaired either, not with today's technology. But here I just want to tell you the method that people use to describe the different cells, the different cell types, or different stem cell types in the spinal cord. I guess I emphasized quite a few times about linear tracking. I have slides showing the manipulating embryonic mouse embryos. In that lecture, I have a similar slide, not exactly. I have a similar slide to tell you what is linear tracking. But here, I want to describe the linear tracking that people used in studies in spinal cord, so you can refresh that. As I said multiple times, this is one of the most important contents I want you to know very well. So in linear track, what people do is you use different promoters to drive Cre. Sometimes it's a modified version of the Cre. So you use different promoter, and these promoters will be specific for certain cells. For example, like the FOXJ1 promoter will be specific for a ependymal cell. Well, CX30 promoter will be specific for astrocyte. Well, OLIG2 promoter will be specific for oligodendrocyte progenitors. So you can predict that the FOXJ1 is not going to be expressed in oligodendrocyte progenitors. They are only expressed in ependymal cells. Therefore, this Cre, the Cre or a modified version of Cre will be specific for a certain cell type. And now you cross this animal with another line of animal, and in this other line of animal, what you have is a universal locus called ROSA locus, driven the expression of a reporter Sometimes this reporter is a fluorescence protein, sometimes it's beta-galactosidase. So this are just reporter, but usually this reporter is not expressed because you have a stop signal before the coding sequence. But the Cre is a recombinase that will be able to cause recombination, therefore remove this stop signal. Therefore, in this locus, you have a recombination. And through that recombination, the reporter now will express because the stop signal is removed. And then the expression is limited to certain cell types, like the ependymal stem cell, the ependymal cells. Why? Because only in those cells you have Cre activity. Therefore, only in those cells you have this recombination. In other cells, you don't have this recombination. Therefore, the reporter won't be expressed. So this is the principle of lineage tracking, because this recombination is kind of a permanent recombination. It won't get reversed. Once the combination occur, you remove this stop signal, they will never get this stop signal back. It's not possible. So this technology will allow you to label one cell and also the daughter cells. The daughter cells of the daughter cells basically permanently label that lineage. So that's the principle of lineage tracking. And now I want you to see the design of the experiment. People want to see after injury, if there is stem cell, how the cells will proliferate and migrate. So you want to track those cells. This is a typical experiment design. In this design, they take the transgenic, that is the result of this crossing, and then they give tamoxifen. Inject tamoxifen. Tamoxifen is similar to estrogen receptor, to estrogen E2. It's an agonist of estrogen receptor. Therefore, this drug will be able to activate the Cre. That's the additional layer of the technique. So with tamoxifen, as I said, that Cre is a modified version of Cre. It has a ER, this estrogen receptor coupled to it. Therefore, the ER will sequester that Cre recombinase on the surface, on the membrane, nuclear membrane. It won't enter the nucleus. Then it cannot cause recombination, because recombination is in chromosomes. It's in the nucleus. Now it can't even enter the nucleus. But with injected tamoxifen, it will make this Cre fusion protein activated. Now it can enter the nucleus to finish the recombination. So this injection is just to activate the Cre. And now the cells get labeled, and now you give an injury, you give a spinal cord injury or without injury as your control. And then next, after a few days, you can take tissue biopsies, or you can isolate individual cells and put them into culture and study self-renewal and differentiation, right? So this is the experimental design there. So what they found is what's shown here on this slide. So basically the conclusion is on the left column, what you see is in the spinal cord, you have three main cell types. One's ependymal cell, others are astrocyte and oligodendrocyte progenitors. So this is very similar to what you found in the brain. And this is the section here shows you that the astrocyte is the dominant cell in normal situations. They are the dominant cell. And then followed by oligodendrocyte progenitors, the blue cell, right? And then the list will be the ependymal cell, because the ependymal cell will be the cells that surround that spinal cord cavity, right? So you have a cavity there, so this will only be present around that region. That's the ependymal cells. This is what you find if you cut a section. And then in the middle column, what it shows to you is the capacity of cell proliferation. So it is kind of hypothetical, okay? So what's shown here is the oligodendrocyte can proliferate. So they can become mature oligodendrocyte. So they have a lot of capacity of replication. So you can see, the potential of this oligodendrocyte progenitor is huge. So you can generate a lot of new cells. Well, the other two cell types, like the red cell, that's the astrocyte, although you find a lot, but their ability to replicate is actually limited. And the ependymal cell will have the least ability to replicate. So they will always have about the same numbers. Well, the astrocyte can proliferate somewhat, while the oligodendrocyte can proliferate a lot. Okay, that's their capacity. Without injury, that's their capacity there. And then with the injury, what happens is, okay, you actually don't have a lot of oligodendrocyte proliferation, and astrocyte won't proliferate a lot either. So what happens is the cells originally surrounding this hole here, that's the ependymal cell, that will proliferate and then give rise to the other two cell types, give rise to astrocyte as well as oligodendrocytes. So they showed that in vivo, in animal, right there, if you create an injury, the cells that surround this hole here, the ependymal cell actually will give rise to the two cell types in this region. It have a lot of amplification and differentiation. And then in vitro, if you take these cells outside the body, and then scientists show that these cells can even give rise to neurons, to give rise to the central commander. So you can tell that in the spinal cord, if you say some cells are stem cells, maybe the cells that some of the ependymal cells are actually stem cells, because in injury they can give rise to oligodendrocyte and astrocyte, but if you take the cells outside of the body, they can even give rise to neurons. So people now are exploring this, as a therapeutic approach, because if you can isolate those cells, maybe you can start to treat spinal cord injury or brain injury. So on the last slide, I just want to tell you a little bit. Although this is a young research area, the neural stem cell field, but they actually have made a lot of progress, a lot more than the field of cardiovascular stem cells. So they actually have a lot of clinical trials already. So they're trying to treat diseases such as spinal cord injury, stroke, and traumatic brain injury. Why people jump ahead and then start to do this? Because as you know, the brain and the heart are super important organs. A lot of people suffer diseases, terrible diseases or injuries because of the damage cannot be repaired, that people lose function, and of course they're not only the quality but also the lifespan will be cut dramatically if you experience some brain damage or spinal cord damage. So there is a need there. So people are starting to using stem cells for treatment. There are human trials, and usually what they do is they either isolated the cell, right? They isolated the cell from animal work, from animal. In humans, there will be some donors, for spinal cord and brain for them to isolate the stem cells. Or you can do reprogramming, like convert fibroblast to neuron stem cells. That is possible now, although we don't cover it, but it is doable. You can do a reprogramming toward neuron stem cells or neurons, or you can induce the pluripotent stem cell or human embryonic stem cell to generate NSCs and then put them into the injury site. And as a matter of fact, actually, some diseases such as amyotrophic lateral sclerosis, ALS, or Lou Gehrig's disease, that's the ice bucket challenge, the disease underlying that activity. Parkinson's, as well as spinal cord injury and stroke, all get treated in clinical trials, and some limited success have already been achieved. So maybe, I would say, maybe in 10 years or so, maybe we start to see therapy based on neural stem cells. Okay, that's the end of today's lecture. You are welcome to stay behind asking questions or feel free to leave.
Actually, I would just have a general question. Why is the basically the neural stem cells known more than the cardiovascular system? Is there a particular reason, like more cases, more studies? Why is it?
That's a good question, but I don't have an answer. I, I, well, I really don't have an answer. It's, they started much earlier.
Mhm.
Altman's work that actually started fairly early, at the same time as the hematopoietic or intestine stem cells. The heart, this start up was late, but I would say, someone will have to bear the blame. The scientist who fabricated data apparently should be held responsible for some of the lack of progress. And then I another thing is, the neuron actually, I don't know, the method in studying these two systems are very different. In the
True.
heart, you can actually do linear tracking, and but the brain system heavily rely on immune histology. You do
Mhm.
morphological study, you don't do a lot of linear tracking. The one reason is because the process is really much slower in other systems.
True.
For the
Yeah.
cells to get mature to become functional, it take very long time.
Okay. Okay.
Yeah, I just don't have an answer for why
Yeah,
the cardiovascular system doesn't have a lot of progress in terms of making use of stem cells.
Yeah. Yeah.
we're actually presenting on that this Friday, so I guess we'll
Yeah. So that's, people are trying hard along
yeah.
along that line, but it's uh, another thing. I guess that applied to both. That's the electrophysiology, right? You could do some therapy, but you could easily mess up the synchronized the beating
Fractions,
in the
yeah.
heart. You can
Yeah.
put
Yeah.
some cells there, maybe, but there is a risk of arrhythmia.
Yeah. Yeah.
Yeah.
So they hesitate to try that in clinical trials. Not in the brain. The brain, if you the chance to cause further damage is not that significant.
I see.
If people is already paralyzed, they probably want to just try, right? So, as long as it's not going to kill people, usually it won't, then you will be, encouraged to. If you see some progress, you will be encouraged to go ahead. But heart, the arrhythmia is a major problem.
That's true. Okay. I see. That makes sense.
Yeah, you put something there, because arrhythmia, the heart is the
Yeah.
good heart, but the arrhythmia can kill people.
Yeah. Yeah. That's the risk is much higher in a sense.
Yeah, I just don't know. I just try to find some reasons.
Yeah. Yeah. I thought like maybe another reason could be like because I know like for instance if you really want to investigate the heart correctly, wouldn't you need to investigate it in flow, like with beating pumps and everything? So compared to like the brain where it's just stands still because you don't need to worry about flow as much because nothing is surrounding it as much.
I guess another reason I can think of is, there are a lot of drugs actually for heart diseases. So basically, you have the beta blocker, you have other things, calcium blocker. You can you can alleviate the symptom.
Yeah, but you
You
won't.
just make the heart to beat slower. You just pump out a smaller amount of blood. Although of course, the function is reduced because maybe the people cannot do exercise or something.
Yeah.
Yeah, that will cause a drastic increase of load, but they can live a relatively normal life. So there are a lot of drugs actually available in the cardiovascular system, as well as devices. Like you can, if some people is experiencing uh, arrhythmia or clot in the batteries,
Yeah.
In vessels, you can put devices in there, right?
Like the balloon to clean.
Yeah. So I guess there are a lot of solutions out there. So when you have a stem cell based solution, when you always get compared to the existing ones. So maybe people will say the risk is not justified,
Interesting. Okay.
right? When you have a lot of the therapy already in place, so the stem cell based therapy won't be accepted by the conventional therapies,
I see. Okay.
right? But in the brain, I don't know. There are
Yeah.
a lot
There's.
of things.
Yeah.
Like some people have Parkinson's. It is really hard to give.
But it's not enough. Yeah.
No, it's not enough. Yeah.
All right.
So
Thank you so
the
much.
help is just minimum compared to what you get if you have a heart problem. The help can be tremendous by other means.
Yeah. Yeah.
Yeah, I just don't know. It's an interesting question.
Yeah. Thank you, Professor.
Oh, you're welcome.
Yeah, I have one question if you have time.
Yeah,
Yeah, do you know any clinical trial for like glaucoma? Let's say we use like reprogramming, like to get some neuron cells, and then we can inject them to optic nerve, so we can find a solution for glaucoma in humans.
I think so. I think that's the area of intense investigation, because this is known that there are stem cells in the eyes, in the ear, in the sensory organs. There are a lot of stem cells, and some of those even use the method we described to identify cells, such as in the ear, they use LGR5. LGR5 as a marker because a lot of the stem cells use, a lot of cells use the same signaling transduction. If Wnt is the driving force to maintain the stem cells, it's no wonder that LGR5 could be used as a marker. Yeah.
Oh.
So I think those are all possibilities. If you're interested, you can read the rest of the chapter. Very, very good chapter.
Because I know doctors say that it's irreversible, and they just prescribe some like eye drops that lower the eye pressure.
Mm-hmm.
So the optic nerve doesn't fade. So if we use like stem cells, we can regenerate optic nerve again, and that's gone.
Yeah, I think even I said that when you have remedies available already, stem cells could make a huge impact if the stem cell are very well characterized and especially if it is safe. Yeah.
Mm-hmm. And one more question: how much do you pay for your mice if you use mice model organism in your lab? Because I was googling, some of them were like super expensive, like $38,000 for just like one specific mutation or transgene mice.
Yeah, let me tell you what. We do our own genomic manipulation. So we put some genes in there. We remove some genes, like I described in our lecture. You can put a cree into a locus, right? We do the manipulation, the genomic manipulation by ourselves in embryonic stem cells. Everything starts with embryonic stem cell. After you made that manipulation in cells, and now you can give it to a facility like Baylor. Baylor have a facility. You just give the cells to them. It will cost you about $2,000. They will give you a mouse.
Okay. Oh.
Yeah, they basically will do the macro injection. They put the cells into the inner cell mass. They mix my manipulated cell to the inner cell mass of another mouse, and now those cells will compete in giving rise to different part of the body of a new mouse, right? New individual. And then hopefully the sperm will carry the cells that I'm manipulated.
Oh.
And then I can pass on that trait in many generations. So that now the line become unlimited, right?
So they give you just give you one mice or just like many?
They give me many because when they do the manipulation, they put into the inner cell mass, but that they will put you into several inner cell mass, not just one. When they do it, it's not when the mouse can carry a lot of embryos. It's not one, not human, only one or two usually, but for mouse, they give birth to 12 or so.
Oh, okay.
So they will manipulate a lot. They put into the uterus of animals of another pseudo mother. So it's going to give birth to a lot of animals, and then from that point on, I do the screening and selection to see which one have the correct manipulation. So it's not very expensive. If I start up the work in my own lab, it's only $2,000. But if you give it as a project to a company, usually they will charge you 20,000 or something.
Wow.
Yeah, then I can't help you.
I was thinking, maybe like how come we don't have like a stem cell lab in our classes? Because I think that's so far. I think that's the best class, that my best science class, along with like immunology, endocrinology, and
Yeah, if
chem.
somebody give me money, I will be able to establish a stem cell lab.
Can we like can we just sign up a petition and give it to the president or Dr. Frankino? I think.
Yeah, I can explore that possibility because it's really interesting. You will be able to culture the cell and make them become hard cell, put in different growth factors, and maybe even start manipulating the cells, like putting a CRISPR and now take out a gene and verify that the gene is taken out. Those are all possible in my lab.
Yeah, but I think so is it Dr. Frankino or who? I think Dr. Frankino is the head of biology lab. I think it's Dr. Frankino. Maybe I asked my friend. Maybe we can start a petition so they can fund you so we can have a lab.
Yeah.
But I think this lab would be better than any other lab. In organic chem we spend like 6 hours. So it's better to spend in one class that's really applied. This is applied science, not like organic chemistry.
I would say that this is, I don't know. I feel good about this semester, although maybe because of the format, online teaching and such, and also myself teaching this for the first time. I just feel like the content is right for seniors, especially for the seniors who's interested in medicine.
Yeah.
And maybe even graduate study. So I feel like this course gives students the opportunity to review, not only to get familiar with the important concept of stem cells, but also an opportunity to review a lot of other things, like cell biology.
Yes. Yes.
Yeah, we use all those knowledge, right? Cell cycle, cell division, the signal transduction.
Yeah, basically this class is giving me like a lot of ideas. Like in like I took immunology, endocrinology, like cell biology. But this is like I think this class comes before them. So you need to know this, then those classes, then you can use that in a lab.
Yeah.
All right, thank you, Dr.
Oh, you're welcome. You're welcome.
Thank you.
You too. So, Joseph, do you want to ask me some question?
Oh, yeah. Sorry, it's echoing a little bit for me. I had a little bit of confusion with the paper for this Friday, more so with the end results, or like towards the end of the paper. When they use the TCF21 tagged cells to reprogram, I just wanted to make sure I did understand this correctly.
Yeah, you can. Which figure?
Yeah, figure 3.
Figure 3. Okay.
Yeah.
Figure 3.
To make sure.
Okay.
So for the GFP ones, it was basically saying that we had like both types of non-cardiomyocyte and cardiomyocyte or cardiac myocyte cells, but they weren't like merging, like they weren't being like changed into it. But for the GHMT, it was going from those like fibroblasts to the cardiac myocytes.
I guess the green part is actually easy to understand because the CTNT is just immune staining, right? Immune staining that specifically mark cardiomyocyte because that troponin T is a marker of cardiomyocyte, not other cells. In the heart you do have other cells, such as endothelial cells, smooth muscle cell, and fibroblasts. So that CT&T will specifically mark the heart muscle cells, cardiomyocyte. So, but I guess what's the most important is the first column. That's the R26R. Let me make it larger so I can see that rest. It is TDT. The R26R TDT is the difficult part. I hope you can explain to the class as well. So that is
That's the roses. Is that like the staining of? I have a note on it. Let me pull that back up because.
No, that is what they did is TCF21 ICRE, right? TCF21 ICRE crossed with ROSA. You see that by that crossing, what they are trying to do is, at the beginning, the cells are fibroblast. Therefore, the cell will express TCF21 because TCF21 is a marker for fibroblast lineage.
Okay.
Right? So now, only in fibroblast, you're going to have a recombination. That's the Cre-LOXP recombination. Through that recombination, the cells will now become red. That's what we reviewed in today's lecture, right? In today's lecture, we reviewed once you have a cell type specific Cre, and then once you cross that animal with a ROSA animal, the reporter will get activated. And that activation is permanent. All the daughter cells now, all the progeny now get labeled as red. So what you can tell now is those cells will, the fibroblast in the heart now will all become red. And later on, no matter what those cells will be, no matter what those fibroblasts will be, they will always be red.
Oh, okay.
Right? Because this labeling is a permanent labeling. Remember that.
So then that's why even
I ask a student to distinguish these two methods, because if you have a fibroblast, you have a TCF21, and now you put red fluorescence directly under that promoter. Okay, consider that. You put a red fluorescence directly under TCF21 promoter. That will only label the fibroblast. Later on, if the fibroblast becomes something else, for example, like become heart cells, it's no longer going to be red.
Okay.
Right? So in that case, if you put a red fluorescence directly under the promoter of TCF21, it will be red in fibroblast, but once the fibroblasts become other cell types, it's no longer red because the promoter is now no longer active.
Okay. So then,
but in this strategy, it's different.
Yeah.
At the beginning, it's already Cre positive, but at the beginning also it is red because of the recombination, right? But later on, even if the cell becomes something else, that recombination cannot get reversed.
So that's why we still see the red in the merged area.
Yeah. I just want to tell you first that you have this concept, even if this become cardiomyocyte, it's still red.
Yeah. Okay.
You see that recombination is like you remove a stop signal from the before that reporter. The reporter is the red fluorescence, right? You remove a stop signal from there, you cannot never gain it back. So when the cell divides to give rise to daughter cells, the daughter cells will always have this genetic mark there. That is the red fluorescence get activated in that lineage. Okay. So now you can track the cell. And now later on, as you can see the merged picture, in the merged picture, in column 3 and also row 3, you see that some cells are labeled both red and green. That the red indicates originally the cells are from a fibroblast.
Yes.
Well, the green means, okay, now the cells are cardiomyocytes.
Yeah.
See, that's so-called linear tracking.
Okay. That makes a lot more sense. I was
Yeah.
sitting there trying to like make sure I understood it correctly.
Yeah, without the green part, you see that the fibroblast now becomes something, but you don't know what that something is.
Yeah.
With the green staining, you know what is a cardiomyocyte.
Okay, cool. And
See?
then I did have, I think,
So you know, you also give you more explanation, like in that merged picture, you see some are only green, not red.
yeah.
That means they're the cardiomyocyte originally in the heart. They are not from a fibroblast. In the heart, you have a lot of cardiomyocyte, right?
Yes.
So only those are both red and green. They are the cardiomyocyte generated from a fibroblast.
So the parts that are in the merge that are just green alone were originally already cardiomyocytes or cardiac myocytes.
Yeah, because in the heart, you have a lot of cardiomyocyte originally.
Okay.
The heart is a collection of cardiomyocyte. And now you make some fibroblasts to become new cardiomyocyte. So this will only be additional cardiomyocyte.
Got you.
These additional ones are labeled red because originally they are fibroblast. From that tracking approach, you can track their origin.
And then I did have for B on this section, I was kind of confused what it was showing us. Is it just the areas that were?
I guess what they're trying to say, because B apparently that's isolated cell.
Yeah. So is it like just section that was damaged after they gave
It's
it?
not a section. It's just the you know, in A is in vivo analysis. Basically, you cut a section.
Yeah.
In A, but in B, you didn't do that. You just take a heart that receive the same manipulation, but you make a single cell suspension of the heart. Essentially, you isolate the cells as single cells, and now they just want to give you a very clean look there.
Okay.
Even if you put the cells as single cells, you see that they are still red, but also they bear a very distinct morphology. That's cardiomyocyte.
Okay.
Because the people in my field, like myself, I know when I see a cell like that, you know, it's like a columnar, and also it shows some striation there. You may not be able to appreciate the striation, but a little bit. So I know it's a cardiomyocyte.
Okay.
So basically, from that image, the scientists just want to tell you, you know, even if we make these cells to become single cells, you see, okay, they're cardiomyocyte, but also they're red. That means they're originally from fibroblast. Because only in fibroblast you have that TCF21 activity. The TCF21 will drive Cre, and Cre will mediate that recombination. That recombination will never occur in cardiomyocyte. So if it is red, that means originally it was a fibroblast. And you do see that in column 3 of B, there are other cells, single cells, right? Other cells, but those other cells are not originally fibroblasts, right? So you do see red cells and non-red cells in column 3, in row 3, in B, right?
Yeah.
So just you can distinguish that some cells that are the original cardiomyocyte, some cells are new and also from fibroblast.
Thank you. I think this definitely helps.
I wanted the students to really appreciate this technology because it is so for modern science, modern genetics, for modern manipulation, using animal as a model. This is the method that people use.
Yeah, I was looking up because I know I had emailed you about who the researcher, like who I should focus on, and I didn't realize he was from, or he goes to, or is a professor at UT Southwestern, or like started the bio department there.
Oh, really? I didn't know
I,
that. He's UC Southwestern. He's a famous professor. He's now the godfather of cardiovascular science.
Because I'm from Dallas, so I I'm in the area, and so when I looked it up, I was just like, oh, that's so cool. He literally did so much.
Yeah, he did so much. He's everywhere. So he has a huge lab in UT Southwestern, and he not only him, his trainees. I mean, his trainees are now well established, big wigs in our field.
Nice. That's so
He's
cool.
kind of like dominated this field. Him and his lineage, his students and postdocs.
Oh, okay, but I mean, it really was a good read. I was struggling a little bit, but I realized that I didn't realize the methods and materials like were summarized at the very bottom. And once I read it, I was like, okay, it makes a little more sense.
Yeah. I hope this. I like this format because I feel it gives the students an opportunity to learn by themselves.
I had to
Yeah,
look.
this is actually very, very important. No matter how well the teacher teaches, ultimately, if the students start to read, and then the effect is tremendous. It's much better.
No, through this class, I know when I started, I hadn't read a lot of research papers. And so it was kind of a struggle trying to look at what everyone was reading and understanding. But it's been easier since your class is very based off of it. So I'm getting used to it and seeing the language and knowing the techniques that you've taught us a little bit.
That's great. That's great. You benefit a lot from doing that.
But thank you so much. I'm going to head out because my group is going to meet to talk about.
Okay.
So.
Okay. Good luck.
Thank
Bye-bye.
you.